So our next speaker is Assaf Amitai from MIT, who's going to tell us about changes in local chromatin organization during the homology search, effects of local contacts on search.
Okay. So I want to thank the organizers for inviting me and giving me the chance to present my work. And this is sort of a data-driven or experimental-driven problem, but once we started to look at it, we saw that we've developed much of the computational power to attack it. So that's a biological question we're interested in. Sometimes the DNA or the chromatin inside cells, in fact it happens on average once for every cell, is going to undergo a double-stranded DNA break. And this can happen when the DNA is exposed to UV radiation, or a free radical is attacking it and just rips the DNA apart. And what happens is that you have those two new loose extremities which are formed, and that's very dangerous for the cell because they can potentially go and interact with other chromosomes, thus creating chromosomal translocation. So this can lead to cell death, the apoptosis, or to cancer in many cases. So the cell has a very complex machinery to repair those damages. And one way to do it is just to religate the two ends. This is called non-homologous end joining. And it's just done by some proteins which come and hold the two extremities together so that they don't drift apart. And then there's a ligase that come and sticks them back together. But the problem with these type of damages is that often you might have a chunk of the DNA which is just ripped off, and then you lose potentially genetic information. Now, how are you going to repair in such a case? One way to do it is to use an homologous sequence, or if you're in S phase and you have another chromosome sitting just next to you, so you can potentially go and use it as a template for repair. And the way it works is that this double-stranded break is going to scan the volume of the nucleus, which is huge. It's, let's say for yeast, the diameter is 1 micrometer, and this is a spot or a locus of the order of nanometers, and then once it's found it, it's going to do what's called a strand invasion, use it as a template for a repair, and disassociate again. So people think that the rate-limiting step for this process is the search, and so that's what we're going to be interested in, how the cell manages to do efficient search for this homologous template. Okay, so people have been studying this process with the following experimental system for a very long time. What you do is you have the mat alpha, on which you can induce a break with an endonucleus, and in close proximity to it you have the lac O repeats, and then GFP connected to the lac I just comes and sits here, and then you have this big bunch of GFPs. We use 256, and this gives you this nice bright locus, and in red you see the membrane. So that's one example. We initiate the break, and then we wait for 2 hours for the breaking efficiency to be very high, so we are quite sure that the DNA has been broken, and we look at this stochastically diffusing point. Now, remember, this is somehow a part of a chromosome, and what we want to do is to try to infer something about its structure and the interaction that it experiences just from its localization. You saw that in the beginning it was peripheral, then it went in, and then it moved again, so something is going on there. Okay, so in 2012, two experimental groups found that following a break initiation, something changes in the way that the chromatin locus moves. So what they really saw was that from this relatively confined trajectory, after you induce a break, it's going to move much more. And at the time, the type of analysis that was done to those trajectory is just calculating the classical MSD. And this is really what people have been doing for a very long time. And we wanted to analyze those trajectories in a more sophisticated manner. So first of all, to quantify the observed change in the locus position, we defined LC, the length of constraint, which is just the standard deviation of the trajectory with respect to its mean position. And we performed, or rather our collaborators, they performed experiments in 2 different time scales, when the time step is 80 milliseconds and when it's 300 milliseconds. And you see that when you induce a break, there's this very strong effect in the change of the constraint length, which is observed in both time scales. So how can we interpret this change in the localization of the spot? Let's say that we are looking at this locus. This is the one with the GFP. But someplace along the chain, there's another locus which is potentially interacting with something. Now, we don't know exactly what it's interacting with. It can be another chromosome. It can be the nuclear periphery. It can be some nuclear organelle, like the nucleolus. But we just hypothesized that there is some interacting locus. So if we look at the typical trajectory of this guy, so we see that it's very confined. And if we look at the trajectory of this guy, which is at a certain distance from the interacting locus, you see that it samples more space. So it has more of a tether than this guy. So the question is, can we, from this trajectory, say something about this interaction and the distance to the nearest interaction point? Okay, so in principle, the problem is even more complicated. Possibly you have more than one interacting potential. And what you can show is that if you look at the trajectory of the blue one, it really depends somehow on the sum of the distances from the nearest interacting potentials, and these guys have a very negligible effect on the way that the blue one moves. So one way to classically extract interactions from a stochastic trajectory is by inverting the Langevin equation. So if we just have a simple Brownian particle moving under this drift, you know that we can have an access to the drift at position x if we calculate the average over all realization of the size of a step in the limit of dt goes to 0. This is just the Carling-Terling formula. And what we want to do was to generalize it for the case of a polymer. Okay, so we assume that we have a Rouse polymer. This is the simplest polymer model, dynamical polymer model that we have. It's just a linear change of beads connected by harmonic springs. And we assume that we have this external interaction someplace along the chain. And you can write down the inversion formula and show that you just have to calculate the marginal, given that RC, this is the observed locus, is its position x. And here we have a gradient over this potential. Okay, so if we take the interaction to be an infinite harmonic well, which is a good first order approximation for any confining interaction, really, you can show that you get this expression for the size step of the observed monomer. And this is somehow reminiscent of an Ullstein-Ulrich process, where x is the distance from the average position of the trap. dc is the diffusion coefficient of the monomer that we were observing, and kc is the effective spring coefficient that this guy fills. And you see that for the case of a Rouse polymer, it has this dependency of, it's inversely dependent on the distance between the observed monomer and the interacting one along the chain, where kappa is the spring coefficient between two adjacent monomers, and k is the strength of the potential, the interacting potential. So from this we can derive an empirical estimator for what is Kc, and we perform some Brownian simulation, and you can show that really in the limit of dt goes to 0, that's you have a very good estimator of this interaction, which is at a certain distance along the chain from the observed monomer. Now the question is, if the localization is due to some effective spring coefficient, and we also measure LC, why should we use this new estimator? And the reason is that often the trajectories that we look at are short, and LC is some sort of a long-time equilibrium measure, while Kc can be extracted from the dynamical trajectory, so you don't have to wait until it sampled nicely all the space. So it's somehow a stronger estimator than LC. Okay, so we looked at several cells, and we could show that really LC and Kc have this inverse relation, as one can imagine, just by extracting those numbers for multiple trajectories. So now we go back to the break problem. We estimated Kc for many trajectories, and you can see that following break induction, you have this very strong effect in diminishing the value of Kc, in both time scales. And the way that we interpret this data is that following a break, the interactions of the potential interaction of monomers around the observed one, and now around the break, is removed, and so this thing can sample larger and larger space. So this makes sense somehow, because we know that once the cell has committed to repair this process via homologous recombination, so some enzyme are going to start resecting the DNA, and nucleosomes are going to be evicted, and if something can potentially mediate protein-protein interaction with other chromosomes or with other things, it might be the nucleosome. So it makes sense that we have this removal of traps around the break site. So we performed some numerical simulation, Brownian simulation, where we have multiple traps on one polymer, and we ask how long it will take for this guy to meet this guy, which is at a certain distance. And not surprisingly, we see that as one removes those interacting sites, the encounter time between those two guys is going to diminish. So you have this direct relation between the number of interacting sites and the encounter time. So we think that that's really what's happening in the physical system, that you just remove those interactions and increase the probability of repair. So what about the anomalous exponent that we might measure for the chromatin locus? So if we take a Rouse polymer, we know that at very short time regimes it's going to move as a Brownian particle. At a very long time regime, the whole thing is going to move again like a Brownian particle with a diffusion coefficient of the center of mass of the polymer. But at intermediate times, it's going to perform subdiffusion with exponent of 1/2. So the anomalous exponent of our locus can be estimated by first calculating the time cross-correlation function. And indeed, you can see that it behaves like a power law, t to the power alpha. You can see the experimental results here. And we know that for alpha equals to 1, this is a Brownian motion. For alpha equals to 2, it's a super diffusion. And for alpha equals smaller than 1, that's a subdiffusive process. We always see a subdiffusive process, but the value of the exponential is not constant. There's a certain distribution of values that we wanted to understand. So when you look at the distribution of those values over the population in the unbroken case and the broken case, we see that following a break induction, we see an increase in the value of the anomalous exponent, and this increase is significant for delta t equals to 80 milliseconds. We still see the increase, but it's not a significant, not statistically significant, at least for 300 milliseconds.
Those are two different experiments.
Yes.
And so the breaks and all the bonds would be different, so there's no reason to. How can you compare them directly?
How can I compare what
When it's one experiment versus
this and this?
Because if you redid the one experiment, you would have different exponent changes and.
Right, right. So we're just looking about what happens to the population. This population with respect to this population. So.
So that's how many experiments in the left?
This is an order of 40, I think,
Okay, sorry.
in both of them.
I misunderstood. Thank
Yeah. So you just look at the population distribution and the population distribution. This is what we did for all the parameters because it's impossible to image and then use the break because of phototoxicity mostly. Yeah, so we see this difference in the distribution. Okay, so wait. One funny thing here is first the fact that we have many cells for which the anomalous exponent is larger than 0.5, and also many in which it's smaller than 0.5. So we asked ourselves what creates this distribution in the value of the anomalous exponent.
Can you give a short explanation what this shape is and what the points, the black points are, and
Okay. So that's called a violin plot, which I newly discovered, and I think it's beautiful. And each of those black points is a parameter estimated for a single cell. So single cell experiment. And then this gives you a measure of the distribution. So there are not so many cells which gave this value, and not so many cells which gave this value. So the bulk of the population is in this regime. Okay.
What is the x-axis actually? What is the scale? What is
So the x-axis, it's not a scale. We have here two experimental situations, unbroken and broken, and these are both wild type.
How do you choose the position of these points on the x-axis?
So that's a technical question about how MATLAB plots a violin plot. So I'm not going to address it now. Okay. Well, the thing is that this is one experiment. This is one experiment. These are the means, and these are the standard deviation. Okay.
Oh, the difference between the numerics.
Okay. So I'll continue.
What was the left and right panel?
Sorry.
What's the difference between the left and right panel? Because the delta.
Right. So these are experiments with the delta t is different. Here the delta t, the time step of the microscopy experiment was 80 milliseconds. And here it was 300 milliseconds.
What is Ks, which is?
Yeah. Okay. So this is a Kolmogorov-Smirnov test to see whether those two distributions are different or not. Okay. So we see. When you see that it's equal to 1, it means that there is an effect. When it's equal to 0, it means that there is no effect. Okay, or at least we cannot disprove the null hypothesis. Okay. So what we did was to label the SPB. This is the spindle ball body. That's an organelle to which all the centromeres of the chromosomes are associated in the East cerevisia, and it's supposed to be sort of a fixed position in the nucleus. And here we have again our chromatin locus. So when you make this experiment, you see that the SPB can potentially move up and down, which is a bit funny because it's supposed to be fixed. So this suggests that possibly nuclear rotation or precession change what we see here. This is one possible artifact of the observation. So we perform a Brownian simulation where you have one end fixed at the origin, and we looked at another end of the chain. We track its position, and we just rotate everything with a certain angular velocity. And what you see, it sort of makes sense because you add a drift term, but you still try to treat it like a Brownian process. If we try to extract the anomalous exponent, we see that it's equal to 0.75, so larger than the 0.5 that we saw. So the point of this slide is that possibly this type of rotations can change or increase the value of the anomalous exponent.
Sorry.
Yeah.
Is it possible to estimate the rate of rotation in a real cell? Because I would expect it should be much slower than the process you started.
Right, but okay. First we don't really know. The only estimate that we have is what I'm going to show now, where we tagged the SPB and we looked at the anomalous exponent and we saw that. So this is some measure of the dynamics. Let's say, but integrated over a few time steps, not just on the, 2 ones, which is the diffusion coefficient. So it tells you if the thing moves with a certain persistence, right? And we see that when we induce a drug, that's a toxin called the troncolin. And what it does is to depolymerize the cytoskeleton, which has a grip on the nucleus and possibly rotates it. So the consequence of this rotation of precession on the nuclear processes is something very new and hot, and we don't really know, but it seems that there is some effect here. So when you remove those motions with the truncaling, really the anomalous exponent goes down. And what's interesting is that if you look at LC for the SPB, so the amount of space that it's sampled, it goes down, but again, it's not significant. So possibly what is happening is that we have this very fast stochastic rotation, but not for very large distances, else it would change considerably the area sampled by the SPB. Okay? So when we look at what happens to our chromatin locus when we use the trunculin, we see that you want to compare what is going on here, and you see that the values of the anomalous exponent, they dropped considerably. So we still have something above 0.5, but it's much reduced with respect to the previous experimental result that I showed you. So it means that we managed to get rid for at least part of those motion artifacts. Okay, but we're still left with anomalous exponent values which are smaller than 0.5. So how can we explain what's going on here? So we developed this other polymer model a while back that we call the beta polymer model. And the idea is that let's consider a case where one monomer doesn't only interact with its nearest neighbors, but there's also some long-range interaction between monomers. And the value of this interaction is decaying with the distance along the chain. So we introduce a new parameter called beta. When beta is equal to 2, this is just a Rouse polymer, just nearest neighbor interactions. So you would have the self-interacting coefficient equals 2. The one with the neighbor will be -1, and the rest will be zeros. But when beta is equal to 1.5, you have those additional interactions. Like I said, it only decays slowly. And we can tune the rate at which the values of interaction decay. So beta goes between 2 and 1. And then we wanted to see what happens to the anomalous exponent of a particle, a tagged monomer, in the intermediate regime. So when beta is equal to 2, this is this curve. We get again the 0.5 that we have for the Rouse polymer, but when beta, for example, is equal to 1.5, we get an anomalous exponent equal to 0.33, which is what you see here. So what we have here is some relation between the observed anomalous exponent of a tagged monomer and some structure parameters beta describing the interactions in the proximity of our observed locus. So that's what it looks like. That's a Rouse polymer really, beta equals to 2. and you see that it has this linear chain configuration. When you start to reduce beta, the whole thing condenses more and more, and that's beta equals to 1.5, which corresponds to alpha of 0.33, which is one of the characteristic value that we obtained in the experiment. And so what we suggest is when you see alpha changes, possibly part of what's going on here is that we have local decondensation. And the reason that we are talking about local decondensation is that we see that the effect is smaller when you take a larger time step. And when you look at a polymer, the time step corresponds to a certain relaxation time. And so you can sort of map the time step to a length scale on your polymer. So that's the value of the distinction between the two time steps. So yeah, this was our prediction that the change in alpha may have to do with changes in beta that would lead, going here, to local decondensation. So we wanted to see if this was true, and what we did was to do super resolution experiment of our system. So like I told you, there is this LACO array here. There are many repeats, and this gives you a certain spot. And what we saw is that after we induce a break in G1, nothing happened, and we know that HR, homologous combination, is not active in this east in G1. However, when we induced it in S phase, we saw an increase in the significant increase in the volume of this spot following a break, which suggests that indeed we have this local decondensation of the chromatin. Okay, so what are the consequences of all of this on the encounter time between two monomers? So again, we performed some Brownian simulation. We have two monomers at a distance r0 from one another, and we want to see what happens when we change beta or change the condensation level of our polymer. And when beta is equal to 2, it's very relaxed, and the encounter, since the probability distribution function of the two monomers are overlapping, these guys are going to meet very fast. But when beta is smaller, this overlap is small. And then what we see is that changing beta, it's a very strong way of controlling the encounter time if one of those guys is bound. And similar to what you spoke about today, when we see this non-monotonous behavior of the encounter time depending on R0, if the two guys are very close to one another, when you increase beta, they're going to overlap less. So this leads to an increase in the mean-first encounter time when you start to relax. Okay, so can we do something analytical with this problem? So we looked at a somewhat similar problem before, the looping of a polymer in a confined domain. So we just wanted to estimate how long it will take for the two ends of this polymer to meet. And since it's very difficult to estimate analytically the partition of a polymer inside a confining sphere, we replaced the confinement with a harmonic potential, and we just calibrated the strength of the harmonic potential based on the end-to-end distance between just the end-to-end distance of the polymer. And so what you get is instead of a Rouse plus reflecting boundary condition, you get Rouse potential plus this harmonic potential acting on all monomers. And this way we were able to asymptotically solve the mean-first encounter time problem and obtain an expression for how long it takes for the two guys to meet, depending on the polymer length, the diffusion coefficient, the capture radius epsilon. This is just how close they have to be for us to say that they've met, and the strength of the confinement or the size of the sphere. So that's what it looks like. You see that when this graph is for the case when there is no confinement, and we know that in this case the mean-first encounter time diverges with the polymer length. However, if we assume that there is some confinement, it's going to converge to some asymptotic value. Its value depends on the size of the sphere. And so Scott yesterday spoke about estimating mean first passage time problems for particles which are performing anomalous diffusion. And in the case where n goes to infinity, the two ends are definitely going to meet before the whole thing has undergone relaxation. So we have a very subdiffusive system. And what is interesting is that we can show that in this case, the process is going to be Poissonian. So there were lots of talks about how can we average out or simplify complicated interaction to enable faster simulation. And the point of this slide is that possibly if you want to simulate a looping event which controls many things in a biological context, you don't necessarily have to simulate the whole polymer dynamics, but you can just find one time the rate at which this happens, and then you just have this type of Poissonian rate of switching from looping on unlooping. So it may be beneficial to greatly reduce simulation time in certain cases. Okay, so we generalize this to the case of two monomers on two different polymers, and this can be solved sort of analytically without Lennard-Jones interactions. So again, this is the formalizing of how we define the mean first encounter time, and replacing again the confinement by a harmonic well, we were able to derive an expression for the mean first encounter time between the two monomers. And what we see is that it depends on the position of the monomer along the chain. For a Ross polymer, since we don't have self-avoiding interactions, the two polymers prefer to be in the center, and that's why the two middle monomers are going to be most localized. And you see that the mean first encounter time is shortest for when you ask middle, and it's going to be the longest when you consider the end and the end. So what you see here is just the result of the simulation, and on top of this I overlap the analytical result.
Are those initial conditions of the two loops, are they somehow equilibrated?
They are equilibrated. Yeah.
In the simulation they would be, but in the calculation, are you?
Yeah. So they're equated. I start from the a long time equilibrium process in both in the simulation and in the analysis. But we can also treat the case where you start, for example, from a straight polymer, because then what you have. So the way that we solve this problem is considering what happens to the eigenspace, eigenvalues, and eigenfunction of the associated Fokker-Planck operator for this problem. And since we can estimate them asymptotically, if you want to take higher eigenvalues which correspond to a straight polymer, we can do it. So, we can do either equilibrium or any initial condition that you have.
What is the small parameter in this case?
The small parameter, it's epsilon. It's the distance when you say the two guys have met. Okay, so what happens if we add the Lennard-Jones interaction? So then this problem changes a bit. And what is interesting is that we see, again, that when we relax the polymer, beta becomes larger, it has a very strong consequence on the encounter time of the two monomers. So this is a very strong way in which the cell can regulate encounter processes inside the nucleus. And then we wanted to study a bit more a specific scenario. So what is probably happening is that following a break, we have some reorganization of the chromatin. We, like I said, we think that we remove local interactions, and this should only change locally what's taking place on the chromatin, at least in the initial stages. So we only remove the long-range interaction for this monomer, where we say that we initiate a break. And what you see here is just the trajectory the color corresponds to time. So you start from red, and you gradually become green. And you see that if we just take a monomer, one of those blue ones, not much is happening. But for the one with the break, it performs this sort of directed motion outside of the polymer globule, and it is pushed to the extremity of the globule. So you can see it if we look at the average distance of this locus from the center of mass of the polymer globule. So initially, before the break, since we took the central one, it was the one closest to the center of mass. But once we initiate the break, it's going to be pushed out very fast and stay in this position. And you can also show that the anomalous exponent greatly increases during this phase. So we wanted to see if we can simulate a scenario of a competition. And first of all, from numerical simulation, you can show that this popping out mechanism is really going to significantly reduce the search time. So this is maybe what is happening. And if you consider the case where you have a break on A and it wants to repair on B, but you have a similar sequence on C, so you're in the danger of doing chromosomal translocation. So obviously when no one pops out, so you have equal probability of recombining of A and B or A and C. But when both A and both B pop out, this is going to bias the encounter probabilities. And the larger the polymer is, the larger this bias on the relative probabilities is going to be. Okay, so this is just a slide to give you an idea of what happens when you start to play with the system and induce mutants in the different repair proteins. So what is interesting is that we see that the respective effect on the different parameters, alpha, LC, KC, and diffusion coefficient, is not the same. So possibly by having this biophysical idea from a polymer perspective on what is happening on the relation between these parameters and the structure, condensation level, interaction, we can start to map the role of those different proteins to the condensation, removal of interaction, and so forth. Okay, so let me conclude. What I showed you here is that first of all, the experimental observation was that following a DSP, the chromatin changes the way it moves, and we can interpret this as some change in its interaction capacity with its environment. And we think that the increased dynamics of a chromatin locus may be related to the condensation, which would in turn significantly affect the mean first encounter time between the break and its homologous sequence. And we can possibly tease out the contribution of those different repair proteins to the modification in the chromatin structure. And finally, this is the confined looping thing that I showed you. We can show that even though polymers have those multiple relaxation times, their encounter times are almost Poissonian in certain limits, both in bulk and in confined domains. And these are all the people who contribute to this work. Most of the experiments were done in the lab of Susan Gasser. Karin Dubrana also did a few, and I work on this with David Holkman. That's it.
Do you expect any difference if the break occurs in heterochromatin versus euchromatin, or is the story basically the same?
No, so there are some papers on this, and we think that the ideas that I showed you are very much relevant over there. Maybe even more relevant when it comes to heterochromatin.
I have two questions, one technical, one more general. How long are the trajectories you measure?
Sorry?
How long the trajectories you're measuring?
Okay, so an order of minutes.
No, how many steps?
How many steps?
The precision also depends on how good statistics you are when you do time average.
Okay, so we started from trajectories of a few hundreds, and now we can have a few thousands also. So, yeah.
And second question, well, you replace the hard walls by a harmonic potential.
Yeah.
Do you know to which extent it's kind of good approximation? How would it affect the results?
Yeah, so I looked a lot at this. First of all, I don't know of any way to do it with hard spheres. So that's one reason to do it with a harmonic potential. But second, when you start to look at the probability distribution function of a polymer inside the confined domain, you see that you have an effect coming from the different monomers. So if we take one monomer and we ask what is the probability of it being close to the periphery, it's not very high, because if it tries to approach other monomers to which it is connected, are probably going to touch the surface before. So in the end, if you look at the probability distribution function of all the monomers inside the harmonic potential, it's all going to be sort of symmetric. And the only difference is that the measure of this confinement is different where you are along the chain. So I would say that a more exact way to do it will be to pick different values of the confining potential for different monomers along the chain. And that will be, I think, a very good approximation.
This may be a dumb question, but do you think there is a chance that when loops form at DNA, that something comparable might be happening? That if the two strands kind of come apart and the place has to find another place to do something, that maybe such reduced times might appear?
You mean so you're asking when we just have to perform looping?
Yes.
Or as enhancer, because enhancers are really far, do we have this type of dynamics or change in the dynamics?
Kind of. Yeah.
Possibly, but I mean, I don't think there are. I don't know any evidences of this. Possibly it can be checked if we do some transcription experiment or something like this.
Okay.
Yeah.
Okay.
Okay.
Thanks, Assaf.